Welcome to another section on Examhood. Today we are looking at, we want to continue from where we stopped last time in biology. Remember last time we looked at osmosis and diffusion. We defined osmosis as a movement of water molecules or solvent from the region of lower concentration to the region of higher concentration through a semi-permeable membrane. The difference between osmosis and diffusion is that in osmosis, there's a semi-permeable membrane. So that was what we discussed last time. So today we are looking at an experiment to demonstrate osmosis using a living tissue. Then what are the materials that are required? The materials include, we need water, we need sugar solution, we need petri dishes, we need yam tubers, then we need knife. And what are the steps or the methods? First of all, you cut the yam tubers into two halves using your knife. Then you make a cavity or a hole into the two yam tubers that you cut. Then you put water into the two petri dishes. You put these yams into the two petri dishes that contain water. Then you label them. The first setup, you label it A, and the second setup, you label it B. Then you make salt or sugar solution. You put the sugar solution into the hole or the cavity that you make into setup A. Then in setup B, you put water. Setup B will serve as the control experiment. Then you leave the experiment for 4 to 6 hours. Then when you go there after that duration, observation now, you observe that osmosis must have taken place in setup A. Remember that setup A, you put sugar solution into the cavity, into the hole that you made in the yam. But setup B, you just put water, ordinary water. So in setup A, osmosis will take place. There will be a movement of water molecules or solvent from the petri dishes. The water molecules will move from the petri dishes through the semi-permeable membrane, which is the yam tissue, into the what? Into the sugar solution in the cavity. So there will be a rise in the level of sugar solution in setup A, while in setup B, the level of water will remain the same. That tells you that osmosis have taken place in setup A, while in setup B, there was no osmosis. You understand? There's no osmosis in setup B because there's no difference in concentration. The two solvents there are water inside the cavity and water in the petri dish. So there will be no movement of water molecules. But in setup A, there will be movement of solvents or water molecules from the region of lower concentration to that of the higher concentration. So in this experiment, the yam tubers, they are serving as living tissues. So this is an experiment to demonstrate osmosis using a living tissue. I hope the lesson is understood. If you have any question, you leave it at the comment section. Thank you so much. See you in our next session.